Gene therapy. What is gene therapy? It is an experimental technique which is used for correcting defective genes that are responsible for the disease development. So, we are doing this by inserting a normal gene to replace an abnormal gene. So, there are also other approaches that can be done. Replacing a mutated gene that causes disease with healthy copy of a gene. Also, inactivating or knocking out a mutated gene that is functioning improperly. Also, introducing a new gene into the body to help fight diseases. There are various researches that are used to study gene therapy for a number of diseases such as SCID, severe combined immune deficiencies, hemophilia, Parkinson's disease, cancer, and HIV. There are two types of gene therapy. One is somatic cell gene therapy and germline cell gene therapy. So, somatic cell gene therapy. So, these therapeutic genes are transferred into somatic cell. Introduction of genes into bone marrow cells, blood cells, skin cells. These are not inherited to the later generations. The other type is the germline gene therapy. These therapeutic genes are transferred into the germ cells. These genes are introduced to eggs and the sperms. These are heritable to the next generations. Gene therapy is further divided into in vivo gene therapy and ex vivo gene therapy. In vivo gene therapy, there are like two delivery systems that are used. One is non-viral and viral systems. So in non-viral systems, there are like nanoparticles that can be used for the gene therapy. In viral delivery systems, viruses as a vector are used for the gene therapy. In ex vivo gene therapy, the cells are isolated from the patient, then they are modified in vitro culture, then these cells are injected back into the patient. The first gene therapy was to correct a deficiency of an enzyme called adenosine deaminase, that is called as ADA. This is performed on a 4-year-old girl, Ashanti De Silva. She was suffering from an SCID, that is severe combined immunodeficiency. This is caused due to a gene coding for ADA. Deoxyadenosine accumulates and it destroys the T lymphocytes. This disrupts the immunity and infectious diseases are caused and she dies at an young age. In vivo gene therapy. Direct delivery of therapeutic gene into the patient's body carried out by viral and non-viral vector systems as we have seen in the picture early. So it can be the only possible option in the patients where the individual cells cannot be cultured in vitro sufficient numbers, that is the brain cells. In vivo gene transfer, it is very necessary when cultured cells that cannot be reimplanted in the patients effectively. In patients with cystic fibrosis, a protein which is called as cystic fibrosis transmembrane regulator CFTR, it is absent due to a genetic defect which is caused by mutation or chemical factors or some other. In the absence of CFTR, the chloride ions are concentrated within the cells and it draws water from the surroundings. This leads to the accumulation of a sticky mucus in the respiratory tract and lungs. This is treated by in vitro replacement of defective gene by adenovirus vector. Vectors in gene therapy. To transfer the desired gene into a target cell, a carrier is required. So such vehicles are called as the vectors. There are like two types. One is viral and non-viral. Viral vector systems are classified into retrovirus vector system. This is the recombinant retrovirus that have the ability to integrate into the host gene with stability. This can carry a DNA of size less than 3.5 kb. Replication defective virus particles. Target cell dividing. Adenovirus vector system. Adenovirus with the DNA genome that are very good vectors. Target non-dividing human cell. Example, common cold virus. Some other viral vectors are adeno-associated virus vector. It is a human virus that can integrate into chromosome 19. It is a single-stranded, non-pathogenic, small DNA virus. These adeno-associated virus enters to the host cell, it becomes double-stranded and gets integrated into the chromosome. Other type is called the herpes simplex virus vector, virus that have natural tendency to infect a particular type of a cell. They infect and it persists in nervous cells. Non-viral vector system. Pure DNA construct. Direct introduction of pure DNA construct into a target tissue. Efficiency of DNA uptake by the cells and expression is rather low. Consequently, large quantities have to be injected periodically. Other non-viral vector is lipoplexes. Lipid DNA complexes. DNA construction that is surrounded by an artificial lipid layer. Most of it gets degraded by lysosomes. DNA molecular conjugates. This is another type of non-viral vector system. This is commonly used synthetic conjugate which is called as poly-L-lysine which is bound to a specific target cell receptor. Therapeutic DNA is then made to combine with the conjugate to form a complex. This avoids lysosomal breakdown of the DNA. Other type is the human artificial chromosome. This can carry a large DNA with one or more therapeutic genes with regulatory elements. How these genes are delivered. There are certain methods such as gene gun method. This employs a high pressure delivery system to shoot tissue with gold or tungsten particles that are coated with the DNA. Conjugation method. Bacterial conjugation is the transfer of genetic material between bacteria through direct to cell contact or through the bridge-like connection between those two cells. Electroporation method. Electroporation is based on the principle that are high voltage electric pulses that can induce cell plasmal membranes to fuse. Thus, electroporation is a technique involving electric field mediated membrane permeabilization.